our Save Our Sisyphus Challenge proposal. As similar study proposals were made by colleagues at both Yonsei University and the University of South Australia, we've collaborated to merge our proposal of the study, is fluoroquinolone use really associated with the development of aortic aneurysms and aortic dissections? So fluoroquinolones are broad spectrum antibiotics, and they can be used to treat a variety of infections, including urinary tract infections, gastrointestinal infections, bronchial infections, including pneumonia, skin and skin structure infections, bone and joint infections, prostatitis, eye infections, septicemia, and also ear infections, including otitis media. As they can be used for so many different conditions, the consumption of fluoroquinolones around the world is increasing. And this is likely due to the resistance to other narrow spectrum antibiotics that are used to treat the same conditions. Thankfully, fluoroquinolones are generally well tolerated and patients generally only experience mild adverse effects. However, over the last decade, post-marketing studies have identified an increased risk of neurological and cardiovascular adverse events following the administration of the quinolones, and they have also reported an increased yet rare risk of aortic aneurysms or dissections. An aortic aneurysm is when the aorta bulges, and aortic dissections are when the layers of the aorta split, which means that blood can leak into the spaces between those layers. And both of these can have catastrophic consequences for the patient. And ultimately, if that aorta ruptures, internal bleeding can occur, which can quickly be fatal for the patient. Another point to add is the fact that the pharmacological mechanism of how fluoroquinolones cause or contribute to these outcomes is not very well understood. Despite this, international regulators, including the FDA in the United States, they've responded to the evidence in the literature by applying these black box warnings and also limitations to the prescribing of fluoroquinolones, and I guess restricting their use to more severe infections where other antibiotics are not appropriate. If I expand just a little on the evidence which these regulatory warnings were made, a quick review of the literature suggests that the quality of the evidence underpinning that association between fluoroquinolones and the risk of aortic aneurysms or dissections could be classified as moderate. So a meta-analysis of observational studies from 2020 identified that fluoroquinolone users had an increased risk of aortic diseases compared to those who used other comparator antibiotics. However, it was noted that there were inconsistencies in the study designs. So of the 5 studies, 2 were cohort studies, 1 was a nested case control study, 1 was a case crossover study, and the other was a case no case study. Interestingly, one of the articles did not report follow-up duration, and some only looked at 60 days after exposure to the quinolone. Also, only 3 of the studies implemented propensity score matching, and other potential limitations of the studies that were included were that the average age of patients in most studies was over 50 years of age, a population who are already at increased risk of these events due to their health deterioration. People with aortic diseases were also often not included, and timing of the fluoroquinolone consumption was not mentioned in most studies. And so as a result, it was difficult to determine whether there was a temporal link between fluoroquinolone consumption and development of their aortic aneurysm or the dissection. And also not all studies reported on individual quinolones, rather that I guess they focused on identifying a class effect. Several other studies have been published since the 2020 meta-analysis. Both positive and negative associations have been reported, and often researchers have listed confounding by indication and surveillance bias as issues that have affected the analysis of this association. So because of the conflicting evidence and the issues with previous study designs, this has led us to consider the following 2 questions, which we hope to answer with your help. So the first is, does exposure to fluoroquinolones actually increase the risk of experiencing aortic aneurysm or dissection within 1 year of starting the fluoroquinolone? And the second is, for a patient with UTI or pneumonia, so a selected condition which the quinolone is indicated for, what is the probability that the patient will go on to have an aortic aneurysm or dissection following initiation of a fluoroquinolone? So I guess what, if any, is the added risk of the fluoroquinolone? So by putting the questions into the framework, we can see that the goal of this project would be to better characterize, estimate, and predict the risk of aortic aneurysm and aortic dissection associated with fluoroquinolone exposure. In terms of characterization, the main thing that we want to do is determine the incidence and time to event of aortic events following quinolone exposure. And so to do this, we can expand those two questions that I just mentioned before to include these two additional questions. So among patients who are new users of fluoroquinolones, how many patients experience aortic aneurysms or dissections within 1 year of initiating treatment? And amongst patients who are new users of fluoroquinolones, what is the time to event distribution between exposure and aortic aneurysm or dissection? So is there a temporal association, and how quickly do these events occur? In terms of estimating the risk of aortic aneurysms and dissections with fluoroquinolone exposure, we also want to compare the safety of quinolones to other antibiotics that are also used to treat the same infections. And we also want to compare the safety between fluoroquinolones to see if there's a class level effect, and also whether individual fluoroquinolones are more likely to be attributed to these outcomes. So again, some further questions we'd like to address are, does exposure to fluoroquinolones increase the risk of experiencing aortic aneurysm or dissection within 30 days, 60 days, 90 days, and 365 days of initiating treatment? And does exposure to fluoroquinolones have a different risk of experiencing aortic aneurysm and dissection within the same time periods of initiating treatment relative to other antibiotics that might be used instead, I guess, to treat the same infection? So to answer these questions, we'll create two pairwise cohorts. These cohorts will initially be divided into patients who are being treated with either a quinolone or an active comparator for community-acquired pneumonia, or they might be being treated for urinary tract infections. We'll also further divide patients within these cohorts as being treated in an outpatient setting or in a hospital setting. And the reason we'll do this is often infections that are being treated in hospital are considered more severe, and we just want to make reasonable adjustments to try and account for things like indication bias or surveillance bias, and also to, I guess, adjust the effect of other confounders as much as possible. As you can see from the table, quinolones and active comparators can be added or deleted depending on which quinolones are available in each data source, and also based on guidelines relevant to the jurisdiction of that data source. Lastly, we also aim to develop a risk prediction model that could be utilised by physicians in the future to assist in deciding whether a quinolone is appropriate for their patient. So the overarching question here is, for a given patient who is a new user of fluoroquinolones, what is the probability that they will have an aortic aneurysm or dissection within a year? So part of answering this question, we'll be able to further identify other risk factors that contribute to increased risk of these outcomes. And I think the most exciting thing about answering this question is that so far, a data-driven patient-level prediction model to estimate the probability of these outcomes following exposure to the quinolones hasn't yet been designed. There is also scope to explore other outcomes of interest as part of the study. Kim, an initial collaborator in this project and infectious disease physician, has suggested that we also further explore things like arrhythmias, mitral and aortic valve regurgitation. We could revisit the association between Achilles tendonitis and tendon rupture. Can also look at retinal detachment, changes in blood glucose levels, seizures, and peripheral neuropathy. Again, each of these outcomes do have reported associations by international medicine regulators. However, utilisation of the common data model via the Odyssey network would allow for better characterisation of the risks of these outcomes with the use of fluoroquinolone antibiotics. So in summary, the importance of answering this question really comes down to the fact that previous studies have had conflicting results and conclusions, yet these outcomes are very serious for patients. Additionally, the generalizable findings in the meta-analyses and the systematic reviews have been limited by the previous study designs. Also, because these outcomes are so rare, randomized controlled trials are unlikely to uncover significant findings, and as such, leveraging the common data model via the Odyssey distributed network analysis is really important to better understand these associations. Collaborating together on this project will also allow for improved exploration of the association of aortic aneurysms or dissections with fluoroquinolones in the primary studies. And the common data model will allow for improved statistical power through larger data sets, which will, in the end, allow for greater generalizability of the findings. We'll also be able to better characterize the time frame in which these aortic events occur following the initiation of fluoroquinolones. And we'll also be able to analyze effect estimates in different age ranges, which, remember, is really important because many of the previous studies focused on elderly populations. As mentioned, there's also scope to create a risk prediction model, and overall we can enhance the evidence on which medicine regulators base their warnings and their recommendations regarding the risks of these medicines. So we're really looking forward to working on this project, and it would be great to collaborate with you and the wider Odyssey network. You can contribute to this study in several different ways, depending on how involved you'd like to be. Some ways that you might like to get involved are by participating in the study design and the protocol development, or you might like to be involved in executing the analysis in your data and then sharing the results with collaborators. There is also the opportunity to assist with authoring publications that arise from the distributed network analysis. And lastly, on behalf of the initial collaborators at Yonsei University and University of South Australia, I'd like to thank Odyssey for the opportunity to share our project idea as part of the Save Our Sisyphus Challenge for 2023. And we look forward to working on this project with you, hopefully very soon.